Hey, well, so you've heard a talk already today from Dr. Frankovich. She kind of gave a good overview of kind of the disorders that we're interested in. So I'm in the lab of Dr. Madeleine Cunningham, and she has done a lot of work and has been very interested in how group A strep can induce autoimmunity that can also affect the brain and cause some of the disorders that we're interested in. And so what I'm interested in, what my work is involved in, is trying to basically try to help characterize and understand some of these potential underlying immune mechanisms that may present in autism as well. And so I also want to thank the Brain Foundation for their support to be able to be here today and for your interest. So hopefully I can provide another interesting perspective as we kind of look and kind of get in the nitty-gritty of trying to understand how infections and inflammation and basically the pathogenesis of and mechanisms of how infections can cause autoreactive responses, and particularly autoantibodies, and why that may be important. And so here I'll just briefly show, as I'm sure some of you have already or been shown, that definitely the prevalence of these or at least awareness of how the immune system can influence behavior is definitely increasing. And so that's something I'm definitely interested in, and to really appreciate how an infectious pathogen can induce a wide variety of disease manifestations. Group A strep is a model organism for this. And we're still trying to understand why. And so group A strep is notorious, or most commonly known, to cause pharyngitis or sore throat. It can also cause a variety of other more rare and more severe complications, such as skin infections, impetigo, cellulitis, septicemia, necrotizing fasciitis. And it also can induce a variety of immune complications where they can cause immune complexes and even cause damage to the kidneys. And then, as if you were here for Dr. Frankovich's talk, where she kind of briefly went over rheumatic fever, is I know, and sadly it's still thought that acute rheumatic fever is a disease of the past. Well, it is coming back. It is still seen in the US. It is still widely problematic in other countries across the globe. And these numbers are very outdated. These are from old publications back on our government websites. So this I don't even know how accurate these numbers are. But we do know acute rheumatic fever can manifest in a variety of ways. It can cause arthritis, carditis, and then Cunningham chorea, which is the most major neurologic manifestation of that, which I'll get into briefly again. And then also it can cause very severe complications of the heart and cause rheumatic heart disease, which can also be very devastating. And then more recently, and still controversial and largely misunderstood, is and a disorder I'm very interested in, is basically the neuropsychiatric complications that can arise following infections such as group A strep, or in PANDAS case it is group A strep. And so really a lot of our work in our laboratory is also interested in trying to understand the similarities with rheumatic fever and RHEA, which is they're very similar in the fact that they both have very prominent emotional and behavioral disturbances. However, Cunningham chorea was likely mostly recognized, at least historically, due to the severe movement abnormalities and the muscle weakness. And so, and it can also have a variety of the neuropsychiatric symptoms that can also manifest in PANDAS, and which most notoriously is mostly associated with this acute onset, where you have a normal, healthy child, but then all of a sudden they just start to regress, where they have these dramatic onsets of OCD and tics, and then they can start to develop these other very severe and drastic behavioral or emotional type of symptoms, that I've listed a few here, but it's not limited to this, such as behavioral and cognitive regression, anxiety, ADHD, sleep disturbances, and even bedwetting. And so just to, and I will get into our work on autism, but I'm really kind of focusing on this so you can kind of understand the mechanisms and how it may actually help in treating kids that may have these similar mechanisms. And so what can happen is, so if you're already an immunologist, or I'm sorry, I'll go over briefly, but you can get these, following infection is you start mounting these innate adaptive immune response, and group A strep can be very inflammatory. It really can activate the immune system, and then it can also mount this adaptive immune responses that are supposed to help eliminate the pathogen. And so it takes about a couple weeks to mount an effective adaptive immune response. And so you can get these cross-reactive T cells that can attack the tissue, which is known to occur in rheumatic fever and chorea, and now we're recognizing in pandas. But you can also get these autoreactive B cells that can generate these cross-reactive antibodies that can target host tissues too, and actually enter into the brain and target the brain as well. And so a lot of the work from my mentor has helped recognize, or she's been very interested in studying group A strep and understanding how it can cause some of these autoimmune responses, as she recognized that there's this phenomenon known as molecular mimicry, where the group A strep can express antigens on its cell wall that for whatever reason, and it's actually a kind of a mechanism for the strep to kind of invade the immune response, because it can generate antibodies, for example, against the group A carbohydrate or M protein, but also it tries to hide from the immune system because it can kind of mimic some of our own antigens. But unfortunately, what can also happen is if the antibodies are happen to cross-react with host tissue and recognize certain autoantigens, which we've identified a few to be important in chorea and pandas and pans, is it can target certain brain antigens such as lysoganglioside, I don't know if I have a pointer here, that's expressed on neurons, the dopamine receptors, as Dr. Kovacic has said, Dr. Kabilka has also replicated this. And it can also, and not only are these antibodies getting in the brain, we're really starting to recognize that they are having a functional effect on the cell. So I know in autoimmune disease, you can use it as kind of a biomarker, which I'll get into as well on how this may be helpful. But if they're actually having some type of physiological effect, a pathogenic response, and if they're getting in the brain at high levels, it can be very detrimental. And so we know that they can activate we have a neurological readout, and it can activate the enzyme calcium homogenin kinase 2, and that has also been associated with other neuronal abnormalities and an increase in dopamine as well. So we know these antibodies are having some impact, and we know they tend to target the specific region of the basal ganglia, which we know also is highly abundantly expressed with the dopamine receptors. And something that I've also been thinking about is I'd really like to look and see if these antibodies if patients have the antibodies, if they're reacting with the amygdala as well, since we know it can play a role in autism and potentially in these disorders as well. And so, since we know based on human studies and animal models that, and even historical studies that have studied in Korea, they just find antibodies that tend to target this region, and there's also inflammation associated with this region. It's interesting on how it's involved, or dysfunction in this region can be involved with autism and these overlapping neuropsychiatric disorders, just due to its involvement with the motor abnormalities and control of the repetitive behavior. And as I mentioned, the potential role of dopamine signaling and the potential involvement of these co-occurring conditions, which we see a lot of the ADHD, OCD, and Tourette's, and pandas and pans as well. And so hopefully we'll get this manuscript out soon, where we really kind of focused on better understanding the role of the dopamine receptor autoantibodies and disease, because we know that they can be elevated in Cunningham, chorea, and PANDAS. And what we're recognizing is that typically D2 is more prominent in acute disease and individuals with Cunningham chorea that have this it's more prevalent in Cunningham chorea, where here we just did ELISA assays where we look at reactivity to the membrane that express the receptors. And then what we're finding with PANDAS is they tend to have autoantibodies to D1. And we investigated a number of cohorts that also demonstrate this. Some of these are very well characterized from Dr. Suedo. And then this was from a really large cohort study with 800 samples of both PANDAS and PANS, and we see the same phenotype. And really just to kind of highlight how this might be a good indicator to help identify at least PANDAS and PANS or chorea or those with this autoimmune basal ganglion encephalitis we did this ROC curve analysis and found that basically autoantibodies to D1 and PANDAS and PANS can predict a reliable sensitivity and specificity for D1, and then D2 for Cunningham chorea. And we also know that these antibodies are not only getting in the brain and targeting the receptors, they actually are influencing receptor activity. These are just some basic receptor signaling assays. There's a lot of work on this. I'm just showing kind of a few data points here where Cunningham chorea serum and monoclonal antibodies derived from patients with Cunningham chorea were incubated on a cell line that expressed the D2, and then we can measure their ability to signal by measuring the second messenger signaling, or cyclic AMP. And so if you know anything about biochemistry, D2 activates an inhibitory G-alpha protein, and so you'll see a reduction in cyclic AMP. And so you see a reduction in dose-dependent signaling with the monoclonal antibody from Cunningham chorea. Sorry, I'm touching here. And that you don't in the non-transfected controls. And you also see this, what's interesting is when you add the sera and measure the signaling, the sera and the monoclonal antibody was able to reduce signaling comparable to that of dopamine, where you don't see in normal controls. And so, additionally, some of my work was with determining whether the D1 antibodies that you see in PANDAS can signal and activate the dopamine receptor 1, because it's the other major class of the dopamine receptors. And we know that they tend to have an elevation of antibodies to this receptor. We saw that the serum was also able to activate our reporter cell line that is actually developed to measure drug agonistic responses. So you also have to keep in mind, you may think that these signaling may be marginal, because as we mentioned, normal healthy patients can have the antibodies, but remember, these patients have inflammation and a lot of systemic inflammation at higher levels. And so I think what we think is going on is it really has to do with the overall abundance and their ability to get in the brain. And then remember, antibodies shouldn't be binding to our cells in the first place, let alone actually being able to bind in a way to activate the receptor. And that's what we're actually seeing in our PANDAS groups. And perhaps Cunningham chorea or other disorders that actually have a high enough level to signal, and if we increased our cohort. So this is something I also want to measure in some of our autism samples. But here I'll just briefly show that to ensure that the signaling was mediated and to better understand the pathogenesis of the antibodies from the serum, we also derived and developed antibodies so we could have a large quantity to study from patient B cells with acute disease. So this is just showing one of our monoclonals that it was able to induce dose-dependent signaling of D1. And interestingly, not only did it activate the signal, if you add it with dopamine, it can actually enhance signaling, and we show this with other assays as well. I'm just showing one here. So we know that that our antibodies not only are activating the receptor, but they can actually enhance the receptor signaling with dopamine. So you can imagine if these patients have the antibodies in, and it's actually binding and signaling the receptor where there's dopamine, how it can cause a lot of dysfunction. And so here is just showing a summary model where we know that for whatever reason, individuals that are susceptible to the group A strep, in a sense that they develop these cross-reactive antibodies that tend to target this brain region, if they have antibodies to D1 or PANDAS, they can activate the receptor and influence its downstream, or its signaling, and potentially maybe leading or exacerbating some of these symptoms. Whereas we know from previous work, D2 antibodies from Cunningham, Korea can influence that receptor, and as well as cause excessive dopamine release, and it has been associated with core-prime movements and symptoms. And so this has really led to our hypothesis in that perhaps we can better identify children with ASD that may exhibit the subtype of autoimmune basal ganglion encephalitis, as you all know, and as the talk from Dr. Fry, it's heterogeneous, and every child may have different symptoms, but if they actually have this underlying mechanism of disease, this is treatable. There's hope, and hopefully this can help them. So we're definitely interested in understanding the role of infections and potentially exacerbating the symptoms as well. And so here I'm just showing some work that went into this manuscript we are working on as well. I'm hoping to get out soon. And I just really want to acknowledge the individuals who have been working on this, as well as Brian Richmond, who sadly is no longer with us, but has made contributions to this study as well. And then also we want to thank Dr. Richard Fry and Suedo has provided samples for this study, and then samples from some of his regressive autism studies to see if we can try to determine if infections and autoimmunity are playing a role in some of those regressive cases, as well as associate that with some of the symptoms. And so we really wanted to compare, so we kind of used PANDAS and PANS as our control group, and compare ASD cases that also had PANDAS, as well as our ASD only group. And we ended up, we did differentiate them into two ages to see if it manifests differently from the younger and the older. And as I mentioned earlier, we know that a lot of the autoantibodies that target the autoantigens can actually activate this neuronal enzyme, CAM kinase 2, which is known to be associated with a variety of functions in neurons, such as synaptic plasticity, dendritic spine morphology, neurotransmitter release, and just a variety of cell signaling pathways. And what we see in PANDAS cases and Cunningham Korea, especially acute and even chronic, is that CAM tends to be elevated. And so that's what we see here with our PANDAS-PANS cases in both age groups, basically in all our ages. All our groups investigated, we see an elevation of the patient's serum to activate CAM on neuronal cells, as well as ASD with PANDAS. And then ASD, its only group, is also significantly elevated, but not to the level of our PANDAS and ASD PANDAS. Same thing with the older group. And so, to determine if the signaling was truly mediated by antibodies, we wanted to actually remove the antibodies from the serum before we measured the ability of the serum to signal the cells. And so we removed them by absorbing them with anti-IgG beads, and we were able to abrogate, sorry, this is really hard to see. We were able to abrogate the signaling of CAM, suggesting that the antibodies are mediating signaling, and then this is just our bead control, and we were able to reduce the signaling as much as our normal sera. And we saw that with both of our serum from our PANDAS and ASD PANDAS groups. And so furthermore, I know I'm going through this pretty quick and this is a lot, but I just want to point out that, as I mentioned, that so now we have antibodies we know antibodies are activating the CAM, but we wanted to see if, since what we know from rheumatic fever, Cunningham Korea, and PANDAS is that you can get these certain antigens from the strep molecule can actually cause this cross-reactivity. We wanted to see if the immunodominant antigen from the group A strep cell wall, which is we coined glucnac, could inhibit the signaling, and that's what we see. So and in fact, our group A strep carbohydrate antigen, and then we chose 2 other common microbial polysaccharide antigens that kids are exposed to and frequently can get sick with. We're also able to inhibit signaling. So it's possible that the antibodies that are causing some signaling could be also react with those pathogens as well. And then here we also wanted to see if our neuronal autoantigens inhibiting signaling to determine if that was that's kind of an indirect way to measure if those are kind of responsible for the CAM signaling, and those were able to inhibit the signaling. So we know with these autoantigens, there tends to be a lot of polyreactivity with the antibody, to where if they're binding one autoantigen, they might actually recognize some of the others. And we see this from both our PANDAS PANS and our AST PANDAS groups. And so, since we know the autoantibodies are present in these groups, we wanted to see if and maybe signaling the CAM, we wanted to see, well, what autoantibodies are actually present in the groups. And so, consistent with the cohorts that I investigated, and what we see is we saw an elevation of D1 in our PANDAS PANS groups, both age groups. We did see some in the young ASD with PANDAS groups, and then we saw some in the older ASD groups, whereas D2 was only in this D1 group, or was in the PANDAS PANS group, and then the younger ASD PANDAS. And then out of the other autoantigens that we see in kind of recognized in this basal ganglia encephalitis is that we saw an elevation of tubulin in the ASD PANDAS group, and then the elder PANDAS PANS and ASD group, but we really didn't see antibodies against lysoganglioside, which we can see in PANDAS PANS. And so with these ASD groups, it tend to be the dopamine receptor 1, tend to be most prominent tubulin, and maybe D2. But to really try to understand, and then furthermore to see, like something we see in Korea and are recognizing in PANDAS, is they tend to have an elevation of immune responses to the group A strep antigen in acetylglucosamine, or this glucnac. And so we didn't see that in our younger PANDAS group, but we did see it in our younger ASD PANDAS group, and then almost in all of our older groups. It would be interesting to see if that would, this plays out in our, in larger cohorts. And this could be likely due to the more common or frequent exposure to infections as children get older, as we are exposed to them. And so then finally, since we know that immune responses generated against the group A strep carbohydrate can induce these cross-reactive antibodies. I know there's a lot here, but it's a lot of the similar data here. We want to determine if these carbohydrate antigens actually could be responsible for the cross-reactivity of the autoantibodies. So we wanted to see if they inhibited, these common antigens can inhibit binding of the autoreactivity. And what we see is in the PANDAS and in the groups that had the antibodies, both the glucnac was able to inhibit binding of D1 and D2, in addition to this Haemophilus influenza type B polysaccharide antigen. But we didn't really see inhibition with this polysaccharide capsule. And another reason I'm showing this is we're kind of finding, and kind of a hypothesis we have with the group A strep sequelae in Korea, is that, and in rheumatic fever, is that these individuals tend to have kind of a thwarted immune response where they develop a different subclass of antibodies, and so they tend to have an IgG2 response, and that's basically it. And so that's what we're wondering to see if that's happening here in these individuals that are susceptible to this autoimmunity. And so lastly, we just want to show, when we did RNA-seq analysis of our sera on these neuronal cell line and to measure the neuronal activity, we were able to see distinct differences and similarities, definitely overlapping differentially expressions between the groups as well, potentially helping us, or just demonstrating that there are shared features in these subtype of individuals that we potentially can recognize and better investigate. And then here is just, again, showing that our ASD groups with PANDAS compared to our PANDAS group, there definitely is similarities in their ability to signal and activate neuronal cells. Sounds like it's time to stop, it's dinner time. With that, I just really, we couldn't do this work with all the funding and recognition over the years. So I'm just recognizing everyone here. As I mentioned again, the Brain Foundation is also helping supporting some of the autism work, and then a lot of work I want to additionally accomplish. And then also, Madeleine wanted to make sure I really recognized and thanked this, I hope I pronounced his name right, Dr. Janicki. He was a generous donor who helped us really get into and further investigate PANDAS and really help identify these autoantigens. So he was, I think, provided some of our first 400 patients to help get that going. And obviously Dr. Suedo, who was one of the initial persons who reached out to Madeleine to really try to understand some autoimmune mechanisms of this. And here's some members of our lab, so I'll be quiet now. Okay.